First of all, I would like to thank Nikolai and the Evocet team for inviting me to present this work. So I'm going to talk about the, is it working? Yeah. About the work we do, which use machine learning and proteomics for bacterial identification in clinical samples. So as everyone knows, antibiotic resistance is increasing all over the world, and it cannot be considered a global threat. And as US CDC claims, a failure to address the problem of antibiotic resistance could result in 10 million deaths per year by 2050 and costing $100 trillion in economic output. So reducing antibiotic resistance is clearly our next global challenge. So the causes of antibiotic resistance are multiple, and we focus our work on the overprescription of antibiotics. So to understand the issues, we have to know the pipeline which is used to identify a bacteria from biological fluid. So usually samples are performed with a bacterial culture and then followed by biochemical tests. And this takes about 100 hours. But during this time, patients receive broad-spectrum antibiotics, which are often efficient to release the infection, but which sometimes are not needed because the patient might be not infected finally. Sometimes they are not efficient and another antibiotics is required. And in all cases, it increases a lot the bacterial resistances in the whole population. So since a couple of years, the biochemical test tends to be replaced by MALDI-TOF mass spectrometry. So it reduces a lot the time to get the diagnosis, but the bacterial culture is still required, meaning that broad-spectrum antibiotics are still provided. So the aim of our work is to replace the MALDI-TOF by LC-MS, which is more sensitive and more specific, and then we can avoid the bacterial culture, and then we can get diagnosis in only a couple of hours. Then the broad-spectrum antibiotics will not be required anymore. So we focus on urinary tract infections because urine is the most common specimen received in microbiology lab, and also because UTI is one of the most common infections. So as you can see here, UTIs are caused by mainly 4 bacteria, especially E. coli, and 15 species represent 85% of all UTIs. So we concentrate our work on these 15 species. And we have initially developed a first pipeline which uses machine learning and to define peptidic signature, able to distinguish between the 15 bacteria of interest. So to do that, we have a first training step where many inoculation replicates are processed through a short protocol of sample preparation and then analyzed in a DIA mode by LC-MS. Then we made a peptide identification through a spectral library, and the data were given to the machine learning algorithm, which were able to give us a peptide signature of 85 peptides, which are presented here. And this peptide are able to distinguish between all the 15 bacteria. So in a second step, these 85 peptides are monitored by targeted proteomics in a non-sample to get the bacteria identification. So this pipeline works quite well because we have an identification in less than 4 hours without culture, with a very good accuracies. But it was one major drawback, which is that the peptide has to be monitored in targeted proteomics, so which need an amount of time. So usually, initially we developed targeted methods with 2-hour gradients, and we reduced that time with run as low as 30 minutes. But even with this time, we can only process 48 samples a day. So then we came with the idea that maybe the peptide identification is not needed, and that we could directly detect the presence of the bacteria in the DIA analysis. So to do that, we developed a new pipeline where we use, again, inoculated urine replicates that we process with the same protocol and analyze by LC-MS/MS in DIA mode. And then we don't make any peptide or protein identification, but just use the raw files that are converted into a format which make us able to make a binning of the signal. So this represents one DIA window, and we can make slices of the signal in 2 dimensions, so the mass dimension and the retention time. And then we get squares where we sum the intensity, and each of these squares is called a feature. So then we can make the same for all the DIA windows, and then we get an intensity table which contains up to millions of features. So these data are then given to a machine learning algorithm, and to be sure to select the best model of machine learning, we use a tool that has been developed in our lab, which is called BiodiscML for biomarker discovery by machine learning. So briefly, this tool works like this. So the data are divided in 2 sets, a train set and a test set. From the train set, the 1,000 best informative features are selected. And then they are tested with different machine learning classifier and different parameters. And only the best feature for the distinction between the different classes, which are here the bacterial species, are selected. Then we obtain a list of models which correspond to an association of one classifier and a selection of feature. And these models are then evaluate on the test set by many multiple cross validations to select the best model for that data. So in summary, this tool allowed us to test 80 different machine learning classifiers with many different parameters and make a feature selection. So at the end, we have tested about 5,000 different models and validate them with multiple cross validations to define the best prediction model. So if this work can take several days, once the best prediction model is selected, it takes only a couple of seconds to get the bacterial identification from an unknown sample. So we have initially validated this pipeline on data we already had on 200 samples from the 15 bacteria representing 85% of all UTI. And they were processed on the Orbitrap Fusion with 2-hour gradients in DIA mode, and we have tested 3 different binning size in the mass and also with retention time. So here you see the data we have obtained. So here are the different size of bins, and here's the number of features that were given to the machine learning, and the model which has been selected with the name of the classifier and the feature which have been selected. So you see that the model select only a small number of features compared to what we have at the beginning. And then is the prediction score that we have obtained, so the accuracy, the area under the ROC curve, and the Matthews correlation coefficient that we like to use to evaluate the performance of our system. And you see that with the smallest size of bin is here that we obtain the best prediction score, which is about 92% of good predictions, with a low error, which means that the algorithm is and the model is very stable. So these features are represented on this heat map, where you see the 35 feature and the 15 bacteria. And here you can see that each feature can be shared by several bacteria, but each bacteria has its own patterns, and it's because of that the machine learning is able to distinguish between the different bacteria. So here we can also see the good performance of the model on the ROC curve, where we have AUC of about 0.94, and the coefficient matrix, which presents the predicted bacterial species here, and here is the truth, so the real bacteria which has been inoculated. So all numbers which are on this line are good prediction, and numbers which are out are wrong prediction, so you see that there is very few. So finally, the idea was to reduce the time of the analysis. So we have now this collaboration with Evosep and with Thermo, and we send them a new batch of samples with 4 different bacteria. And these samples have been run on the Evosep system, coupled to the new Orbitrap Exploris 480, which was running in the DIA mode. And with the Evosep, we were able to test 3 different gradients, making us able to analyze 60, 100, and even 200 samples per day with runs length as small as 5.6 minutes. So we process the data as described before with the machine learning, and you see here the results for 2 binning sites, and you see that with the smallest one, this is where we obtain the best results. And for all gradient, finally, even the smallest one, we were able to get prediction score higher than 80%. So the good performance of the model and the method is also shown on these ROC curves, where we have AUC also above 80% for all 3 gradients. And finally, on the confusion matrix, where again we can see the few number of wrong predictions. So of course, the idea is to improve even more this number, and maybe reach 98 or 99% of good prediction. But there is still improvement that we can do. So we are now working in the idea to modify the number and the size of the DIA windows. We also think that we could include MS1 information to improve the prediction, and we are also working on the retention time binning. Finally, this work will be extended to the 15 bacteria representing more than 85% of all UTI. We will really like also to test the shortest gradient that we can do on the Evosep one, which is 300 samples per day. And we want to also to work on normalization and retention time alignment to avoid batch effect and make the method transferable to other instruments. So as a conclusion, I can show you this little table, which presents the method that we can use for identification of bacteria in urine. So the standard method, the MALDI-TOF, requires a long amount of time to get a diagnosis due to the bacterial culture, but is able to analyze several hundreds of samples per day. So the first method that we have developed with machine learning on peptide identification make us able to really reduce this time to a couple of hours, but we were not able to analyze more than 50 samples a day. And now with this new method, we have again a very good time to get the diagnosis, but we are now able to analyze 200 samples per day. So in the future, we are also thinking to transfer this method to other clinical samples, or also to issues in the food industries. And it will be also very interesting to see if we could be able to detect specific resistances or virulences. So to finish, I would like to thank the people working with me on that project. So in the Arnaud Droit Lab in Quebec City in Canada, so Michael, Marion, and Clarisse, people from the Infectiology Research Center who provide the bacterial strain, and our collaborator from Thermo and from Evosep, especially Tabi and Nikolai, who run the sample with us. So thank you to all of you.
So thank you very much, Florence. This is on. This is not on. Okay.
Oh, sorry. Did you?
I thought it was started.
Yeah, it was okay.
Anyway, thank you very much for a very nice presentation.
Okay.
Do we have any questions?
I think I can just talk. Everybody hears me.
Go for it.
Right. So I was wondering, have you tried this on real patient samples?
We have tried with our previous method with peptide identification, and of course, yes, it's the next step that we have to do with this very short gradient.
And then also are you able to detect the infections of the multiple bacteria, because sometimes they have very often have co-infections, right? So not only a single species.
You mean multiple infections? We have already tried two different bacteria with the first pipeline, and of course, it's also something that we have to try because multiple infection can happen. So
do you see a deterioration of performance when you have multiple coexisting bacteria?
As soon as the machine learning has enough replicates to be trained, there is no reason to think that it won't work. It really defines the model's function of the replicates that we use to train the machine learning. So by the time we can improve all the predictions and the number of bacteria that we could detect by adding more and more data to the model.
Thank you.
Any more questions for Florence? Okay, so please join me in thanking Florence one more time.